Welcome back. Today we have another installment on our ongoing series on Alzheimer's disease. In particular, I like to focus on the notion that perhaps we need to reject the well-established amyloid cascade hypothesis of Alzheimer's disease. As you know, we have done two previous presentations on breakthroughs in Alzheimer's disease, describing how a monoclonal antibody was able to remove a great deal of the amyloid deposits in Alzheimer's patients. And in another presentation, a small molecule was discussed, which is an inhibitor of a protease, which also is able to clear amyloid deposits from Alzheimer's brain, all of which seems to support the notion that the amyloid hypothesis of Alzheimer's disease should reign supreme, and it really has elicited all the funding in the world, and many drug companies have literally poured billions of dollars into researching that particular aspects of Alzheimer's disease. Now today, I will try to undermine in a progressive way the notion that the amyloid cascade is the only story, or perhaps even not the determining story in the pathophysiology of Alzheimer's disease. And towards the end of the talk, we will arrive at a paper which was just recently published from a lab at Stanford, which sheds entirely new light on what might be going on not only in Alzheimer's disease, but in a number of neurodegenerative disorders, such as amyotrophic lateral sclerosis and Parkinson's disease, in addition to Alzheimer's disease. There may perhaps be a common pathway in which all lesions that eventually lead to neurodegeneration end in the activation of what is called M1 astroglia, neurotoxic astroglia, and we will approach this in a stepwise fashion. But first, let's talk about the amyloid cascade hypothesis of Alzheimer's disease. So the way it's classically stated is that over time, an imbalance in amyloid beta production and or clearance leads to gradual accumulation and aggregation of this peptide in the brain, then initiating a neurodegenerative cascade that involves amyloid deposition, inflammation, oxidative stress, neuronal injury, and so forth. So the amyloid hypothesis for Alzheimer's disease posits a neuron-centric linear cascade initiated by A-beta deposition and leading progressively then to tau pathology, synaptic dysfunction, inflammation, neuronal loss, and eventually dementia. Now, if this is all true, then why is there no good correlation between the levels of amyloid beta in the brain and cognitive impairment? In fact, the correlation is quite poor. The evidence overall seems to suggest that there is a long, complex cellular phase consisting of feedback and feed-forward responses involving astrocytes, microglia, and the vasculature as well. So the amyloid hypothesis has no explanation for the silent incubation period of Alzheimer's disease. Even in those cases of familial inherited Alzheimer's disease with early onset, why is it that you have the genetic lesion from the womb and childhood? Why is it that it takes decades to develop the pathology if the genetic lesion alone is the driving cause? So although abnormal amyloid beta production is already in place from conception, the toxicity manifests itself only in midlife. So there's no good explanation that can be derived from the amyloid cascade hypothesis alone. To remind you, here is then a compilation of the various genetic lesions that can occur in the amyloid precursor protein. And as you can see, they come in two varieties. One is the BCE1 side, and the other one is the gamma secretase side. And you can see that there is an increased pathogenicity as you go to the A beta 42 type peptide. There are also protective mutations involved here. So this seems then to suggest that amyloid is really critical and important in Alzheimer's disease. Here now is a way to kind of rescue perhaps the amyloid hypothesis by adding some complications. For example, we know from genome-wide association studies that the ApoE4 allele, either heterozygous, even more so in the homozygous state, is strongly promoting the occurrence of Alzheimer's pathology. So we add this wrinkle over here. We already have discussed the various mutations that can down the road lead to Alzheimer's disease. And we should not forget Down syndrome, trisomy 21, where we have an APP familial mutation that also can lead to Alzheimer's pathology. In past decades, Down's patients did not survive very long, but with improvement in medicine and management, Down's patients can now live into the fourth, fifth decade and manifest increasingly Alzheimer's disease pathology. And of course, the driving force is, as always, the A beta aggregation. A beta comes in different forms, but here we have the A beta 42, perhaps the most toxic. Soluble oligomers and monomers are involved. The amyloid plaque is formed, leading to what's called here the aggregate stress. Now, independent and parallel in a sense is the tau pathology. So hyperphosphorylated tau tends to accumulate, perhaps unrelated in some way to the amyloid pathology. But tau, of course, or the extent of tau precipitation in the brain, has a much better correlation with cognitive impairment than does the beta amyloid peptide. So here we now have a hint of where I want to go with this talk. You see here microglia and astrocytes. In some way, there is an importance to the inflammatory response in the brain to be it an amyloid deposit, be it a toxic A beta 42 oligomer or monomer, or be it a hyperphosphorylated tau. Without inflammation, nothing bad seems to be happening. So there is another hidden layer of molecular events that is not addressed by the simple statement of the amyloid cascade hypothesis. So to remind you here, what we're looking at here is evidence for microglia in an amyloid beta deposit, in brown here in panel A. You see the brown amyloid deposit in the center, and the activated microglia in blue is hovering around the amyloid deposit, getting ready to eat this protein mess up, phagocytose, in other words. In panel B, we have again activated microglia, this time in the brain of a transgenic mouse that is hyperexpressing the A beta peptide. And in panel C, you finally meet the astrocyte. Here are the astrocytes. And astrocytes are just very fantastic cells. Did you know that one astrocyte is involved in nurturing, maintaining, and perhaps, I give the story away now, killing up to 140,000 synapses? So that is the widespread connection that one astrocyte can make in interweaving itself into the synaptic network in the brain. We will come back to this in a little while. Now here in panel D, you see astrocytes again, this time in green with fluorescence, and this is again from a transgenic mouse. And in panel E, you see interleukin-1 beta positive microglia in the frontal brain section of a patient with Alzheimer's disease. So we have known then for a long time that microglia and perhaps astrocytes are involved in dealing with the abnormal protein precipitates in the brain, trying to clean the damage and to have the brain go on to live another day. Now, complications arose quickly when genome-wide association studies were performed on Alzheimer's disease, and as it turns out, there is a host of susceptibility loci in the genome. In this case, 22 are listed here, and interestingly, a number of these loci locate to the immunoreactive complex. In particular, there is a gene called TREM2. TREM stands for triggering receptor expressed on myeloid cells, 2. So this gene is part of the innate immune response. And genetic studies have shown that rare mutations in TREM2 are a risk factor for Alzheimer's disease. Turns out that TREM2 is kind of a control switch, a gateway to the microglia response to injury in the brain. So here's then a panel that shows you where things fit together in terms of the genetic association studies. You can see here rare mutations, and up here are causative mutations. These mutations are the causes of a subset of Alzheimer's disease, namely familial Alzheimer's disease. And you see presenilin 1 and 2, these would be the gamma secretase, and mutations in APB itself. Now here you see something interesting, namely ApoE4. ApoE4 is a very common allele. This is the frequency of the allele in the population. So ApoE4, involved in lipid trafficking and processing, is very common in the population in the heterozygous form. It's much rarer as a homozygous, but if you are homozygous for ApoE4, your risk of developing Alzheimer's disease has increased by about 8-fold. And here you see in a blue box TREM2. So this is then a receptor on microglia that is involved in microglia activation. And it's in blue because it's part of the immune response. You can see here on the right side of the panel a host of other common alleles in the genome, but they have very small impact and carry low but measurable risk. So TREM2 then is a protein that mediates microglia phagocytosis and promotes an anti-inflammatory cytokine profile, with some caveats to which we will get in just a few minutes. So here then is a paper on the TREM2 mutations that have been implicated in neurodegeneration and impaired cell surface transport and phagocytosis. So genetic variants of this gene have been linked to a number of diseases, including Alzheimer's disease, Parkinson's disease, ALS, as well as frontotemporal dementia. So we know a lot about TREM2 up to the structure of the protein itself, and here you see the various mutations that can occur that lead to abnormal processing of this protein from the cell membrane of microglia. And the key finding is here. In frontotemporal dementia, a fragment of TREM2 that is normally released is significantly decreased in the cerebrospinal fluid. So here are your controls, and you can see there's a significant decrease at the 0.001 significance level. There's also a decrease in Alzheimer's disease. So the plot is thickening. In some way, the microglia activation pathway, as assessed here by abnormal TREM2 genes, is somehow involved in Alzheimer's pathology. So just to rub it in one more time, one of the weaknesses of the amyloid cascade hypothesis of Alzheimer's disease is the fact that tangles correlate better with the cognitive impairment than amyloid plaques. Tangles, of course, being produced by hyperphosphorylated tau, neurofibrillary tangles. So then, let's halt and summarize very briefly. There is evidence then to support the view that Alzheimer's disease may present a failure of autophagy or lysosomal function. A strong case can be made for neuroinflammatory events. So an injury has to occur which is distinct from those events that accompany normal aging, and followed by the establishment of chronic inflammatory state and cellular changes of state that permanently alter the biology of these cells. Now, what about just plain old inflammation? I ran across this interesting paper which appeared a number of years ago from the University of Zurich. So these investigators injected pregnant mice with a poly-C model. This is a molecule that mimics a viral infection. So these young moms were exposed during pregnancy to this inflammatory producing molecule, and the offspring were later studied. And as it turns out, there is amyloid deposition in the offspring. Now, these are not transgenic mice. Remind you, mice don't get Alzheimer's disease. They only get Alzheimer's pathology if we insert into their genome abnormal genes derived from the human genome that are then expressed in the mouse. But here you can see that if you have a mouse that is a carrier, a transgenic mouse, 3 times transgenic mouse, meaning it has 3 different genes associated in the human with Alzheimer's disease, and this mouse was exposed to this fake viral stimulant in utero, you see massive deposition of amyloid pathology in the hippocampal region here. So inflammation or infection is probably one of the main mechanisms in which Alzheimer's disease pathology is triggered. I'd also like to refer you back to a presentation that we had entitled, Is Alzheimer's Disease an Infectious Illness? And there is a host of data to suggest that the amyloid protein itself may be able to target invading organisms such as fungi and bacteria, and may play a role in the host defense against invasion of the brain. This is a parallel line of investigation that is pursued in a number of laboratories around the world and should not be totally forgotten as we concentrate so much on the amyloid cascade hypothesis. Here's just another summary of this, and you can see that these mice have the typical deposition of microglia here, but they also have behavioral deficits. If they run in a maze, they become forgetful and are not as good as their non-infected litter mates in finding their way around and learning how to get the piece of chocolate at the end of the maze. All right, let's stop again and summarize what we have learned so far. So we know that Alzheimer's disease is associated with network failure, synaptic failure, cell death, and dementia right here in the center. Seems like there are various ways how to get there. One, of course, is the genetic risk factor way, specifically those genes that have been proven causative for Alzheimer's disease somewhere over here. But then we also have the APB story and the tau story in this area. And now we have other stories, such as perhaps a failure of phagocytosis or a failure of lysosomes to chew up abnormal protein depositions. So you can see how the story is broadening into other areas of cell regulation. Now, some people, when assayed for the presence of amyloid deposits, have a lot of amyloid in the brain, but do not show any clinical manifestations of dementia or Alzheimer's disease. And what is different about them? Well, here is a list of what is different. First of all, they have a striking preservation of neuron numbers. So the synaptic markers are intact, the neurons are still there despite the fact that amyloid is all over the place. Now, the patients with dementia had more oligomeric amyloid deposits and certain specific antibody responsiveness. Also, the accumulation of hyperphosphorylated tau separates patients with dementia from those that have a lot of amyloid and no dementia. And finally, perhaps most importantly, and the key part of our story today, is a robust glial activation accompanying the amyloid beta and tau pathologies in demented patients was remarkably reduced in patients with amyloid burden but without signs of dementia. So you can accommodate and live with amyloid deposition as long as you don't activate the nuclear option, a hyperimmune response that will lead to the destruction of the neuronal network leading to dementia. And we will now slowly approach our key paper and show you what might be the cellular mechanism of this. So again, here we have evidence for a long, complex cellular phase process. Amyloid cascade hypothesis cannot explain significant features of the disease. Which brings us to a cell called astroglia. Now, when I was in medical school or in psychiatric residency, nobody talked about astroglia. It may have been mentioned in passing as one of the boring cells in the brain that may have some kind of structural or supportive role of some kind to brain function. As it turns out, synapses cannot connect and form successfully without the assistance of local microglia. Microglia are it's like a nursery that allows synapses to be successfully implemented. This is the reason why we can call a synapse really a tripartite arrangement. We have a presynaptic terminal, a postsynaptic terminal, and we have an astrocyte sitting right here listening in to the traffic that occurs from the presynaptic to the postsynaptic terminal and intervening in very important ways to make sure that this traffic is maintained and sustained in a healthy way despite a great deal of toxic events going on, such as glutaminergic transmission. So here is a cartoon talking about the maturation of synapses assisted by astrocytes. Astrocytes release compounds called glypicans here, which allow for certain changes to take place in the synapse. In particular, the production of AMPA receptors here, which cannot happen unless we have NMDRA receptors activated. Now, it's very difficult to get NMDA receptors to fire because there is a big fat magnesium ion in the center of the receptor. And what microglia allows is to increase the clustering of the glutamine A1 subunit of the AMPR receptor and therefore induce the functionality of the AMPA receptor subtype. So without astrocytes, there is no excitatory activity going on in the brain. And as I told you before, one astrocyte can service about 140,000 synapses. Now, here is the downside, or perhaps the maturation side of astrocytes, and that is, as you know, in childhood, a great deal of excessive synapses are produced. These are cut back and pruned in adolescence, and you need a responsive and clever mechanism to do this in the appropriate way. So astrocytes then are involved in secreting a signal, which I think is as yet unidentified. However, this signal leads to the C1q complement system expression on the synapse, as indicated here. And this allows the production of C3, which allows for microglia to be activated because it has complement receptors. So that is a mechanism by which astrocytes are involved in the elimination of targeted synapses. Now, there's a very recent paper, we will not talk about it at length today, but it will be one of our presentations coming up, which shows that during sleep, there is wholesale remodeling of excitatory synapses going on. And it's a very exciting finding, which explains perhaps why sleep is essential for learning new skills and retaining new information. Without adequate sleep, you can study all you want. You will not remember. And we now know, or know at least part of the story, as to why this is happening. And it also involves astroglia and microglia. So let's summarize here. Astroglia then are important in synapse elimination, and we know a great deal about the microbiology. Namely, there are certain docking proteins, which are the so-called eat me signal. So we need to tell microglia which structure to chew up, and this is mediated by a combination of C3 complement and certain receptors on the astrocyte. And here, of course, is the complicated story of how astrocytes are involved in neurotransmission and neuroplasticity. We will not go into some detail now. That is a different talk. Okay, let's move on. This is just a larger version of the picture I showed you. Let's move on to the next slide. And here we can see a attempt to compile the various phases of Alzheimer's disease. We talked about the biochemical phase, which involves in some way or another abnormal amyloid beta. We then have a cellular phase, which we now believe involves in some way astroglia and microglia over here. And this cellular phase can take a long period of time to become effective, but eventually does lead to destruction of synapses and neurons. So here is a astroglia cell in action. You can see the protoplasmic astrocyte in green enveloping the cell body, and the processes of a neuron in red. So you see the bushy nature of these astrocytes in the image, and you can imagine the multitude of connections that this astrocyte may be able to make with synapses in its immediate neighborhood. Now here is a YouTube video that you can watch. This was a talk given by Ben Barres. He is the leader of the laboratory at Stanford who put the astrocyte on the map. and he is one of the key authors of this paper that we are approaching now, that shows the existence of neurotoxic astrocytes and their involvement in perhaps all neurodegenerative diseases. This was a very amazing talk. I encourage you to watch this. It was given in Boston. A number of important researchers, including Beth Stevens, were in the audience. Beth Stevens is the person that we have talked about earlier in the genetics of schizophrenia. She is the one that put microglia on the map as a possible cell involved in pathology in schizophrenia. So I encourage you to watch this YouTube video. So just to recapitulate briefly what microglia is supposed to do, how does microglia eliminate synapses? Well, it involves critically complement C3. So complement C3 needs to tag the synapses that need to be eliminated. Here's another cartoon. Another key protein is C1q, another element of the complement system that we will encounter again. Here, of course, is CR3. So this has been known for quite a while. This is the classical complement cascade. Little was known that this was actually occurring in the brain. This is what makes this so exciting. So the classical complement cascade calls for microbes and debris being detected by a surveillance system in the body. It activates C1q first, recruiting C4, C2, and eventually C3, leading to phagocytosis in the end and lysis in the lysosome of the offending agent. So this mechanism, this entire machinery, is also used by the brain to sculpt itself, to deal with the restructuring of neuronal networks, and also on the downside, lead to the destruction of entire neuronal network and dementia. Just as an aside and a reminder, we had to talk about the genetics of schizophrenia. In this case, it was the C4 part of the complement system that was critically involved in the schizophrenic risk. So then this is the paper we want to discuss. Neurotoxic reactive astrocytes are induced by activated microglia. So let me tell you briefly how this happened. This lab was able to purify microglia and astrocyte cells and maintain them in a what's called serum-free medium. So this is a medium that has all the components that the astrocyte need to survive and do its job in a Petri dish. So now you can study the interaction. How do microglia and astrocytes talk to each other? And what this lab found that there are three factors: an interleukin, a C1q, and another factor that are sufficient and are causal in changing an astrocyte from a nursemaid and a benign cell that will support neurotransmission into an absolute killer, which will eliminate synapses and cells. This was shown by so-called deafferentiation. You know that the retina sends the information via the optic nerve to area V1 or the CGN first, but if you crush the optic nerve and go to the retina and examine cells in the retina, you can see that cells are being eliminated by an astrocyte cell that is phagocytotic and has been activated. You can stop this destruction with antibodies to these three activating factors that have been shown in the Petri dish, released by microglia, which will convert a normal astrocyte to a so-called M1 neurotoxic astrocyte. Here then is the paper that just came out in Nature. And you can see here Beth Stevens is one of the authors, and Ben Barris, of course, is mentioned as well. So let's scroll down. In summary, then, what these authors discovered is that you can convert a normal astrocyte into an M1 astrocyte by interleukin-1 alpha, tumor necrosis factor, and C1q complement. Those three and only those three are required. And they show that these three factors are released by activated microglia. And let's scroll down and show you this interesting paper. So this is a way to determine that the astrocyte has been converted from a harmless astrocyte on top of the panel here. And in the middle, you see all the red markers here. This is a astrocyte that has been converted into the toxic form. The astrocytes lose the ability to promote the maturation of synapses, which is their normal function. But what they acquire is a powerful neurotoxic effect. So here you see retinal ganglion cells that have been exposed to this intervention where the nerve connections are crushed, and you can see red dots appearing in 12 hours and on the top panel, and you can see that a red dye is invading the nucleus, which means that the cell has died. So cell survival then goes down in the panel C, D, and F, and this is mediated by the M1 astrocyte. Again, here in panel G, you see the normal astrocyte on the top, or the blue colors predominate. At the bottom, you see what happens to the astrocyte after a nerve crush has occurred and the neuron has been disconnected from where it's supposed to connect in its next synapse. And you can see that this crushing or deafferentiation transforms the genetic expression of genes from this astrocyte, and you see all these red dots here in the middle panel indicating activation of a host of different genes, including IgG, tumor necrosis factor, and C1q. Here in panel I, you can see that the control is no crush, and you can see that after you crush the nerve, you get mass destruction of neurons in the area mediated by the astrocyte. So here is really the key panel. Now, look at this very closely, and you can see that on the top panel, A, B, C, D, and E, you can see 5 degenerative disorders: Huntington's disease, Alzheimer's disease, ALS, Parkinson's disease, and multiple sclerosis. And in one form or another, you can see that C3 expression, indicated by these punctual accumulation in the cell bodies, by in situ hybridization. In other words, looking for the expression of C3 complement messenger RNA in these cells, you can show that the gene for C3 is activated in the cell bodies derived from patients with Huntington's disease, Alzheimer's disease, ALS, Parkinson's, and multiple sclerosis. Here in panel I, you can see that normal controls don't have any C3 in this area. So there is a 60 or 70-fold activation of C3 transcription. The same is true for the other disorders: Alzheimer's disease, ALS, Parkinson's disease. So this is then an amazing finding that would somehow put all these neurodegenerative disorders in one basket, namely that the ultimate cause of death, the ultimate manner of death of the neuron may be a toxic activated M1 astrocyte, which is the final outcome of a pathway through some kind of injury, genetic or otherwise, activated microglia, and then bringing in the toxin produced by the M1 astrocyte. Okay, here is a presentation of the different factors that are released by the microglia. There are 3 of them which are sufficient to promote the M1 astrocyte production. And here's the panel I just showed you in the context of the paper. Now let's go to the next slide, and you can see here, hopefully, the accumulation of these little punctate hybridizations of C3 complement factor. In Parkinson's disease, we use a different technique. Namely, we have a staining, immunostaining for C3. And here, and you can see it's positive as well, and the same is true for multiple sclerosis. So then, what does all this mean for Alzheimer's disease? Well, the causes of Alzheimer's disease may be manifold. We talked about genetic causes, the various mutations involved. There are studies which show that infection or immune response may be involved in Alzheimer's disease. There are studies that show that the blood-brain barrier becomes eroded and porous, in particular around the hippocampal region, and that this erosion of the blood-brain barrier may allow pathogens to enter the brain, which may elicit an amyloid deposition response, which then, of course, may activate microglia, which then may activate astroglia, transforming astroglia into the M1 type, leading to neurotoxic destruction of an entire network. So you can see that although the amyloid cascade hypothesis on the surface is very persuasive and has attracted a lot of attention, grant funding, as well as money from the pharma industry, the story may be more complicated than that. But on the other hand, it may open up new therapeutic avenues. For example, antibodies to IL-9 are available commercially, and perhaps the intervention can take place at this critical conversion of microglia and astrocyte from normal astrocyte to M1. If we can prevent this step with some kind of monoclonal antibody, perhaps the nuclear destruction, the nuclear option that microglia and astrocytes seem to evoke may be preventable. So that is the end of this presentation. We will combine all of our Alzheimer's presentations into one package called the Neurodegeneration Project. And we will make this available in one coordinated fashion. We will let you know when this will happen. And I think it will be quite useful for you to look at all these presentations in a row, as it will give you an in-depth understanding as to where the current thinking is in Alzheimer's disease and other neurodegenerative diseases. Thank you very much, and we'll see you soon again at Behavioral Health 2000.